All right. So thank you very much to organizers for giving me opportunity to attend this meeting. I'm attending such a large gathering of structural physics after a very, very long time. So I'm going to talk about some computational modeling for sliding and clustering of motor proteins that we have recently developed and published, and how it affects the transport. So just to give you a brief introduction about what we do in the lab. So at IIT Bombay, I have a biophysics and bioinnovations laboratory, and there are three different types of problems we study in the domain of biophysics. We study the transport and force generation by molecular motor proteins. Then we also study the interaction of potential anti-cancer drugs and maps with microtubules. And we also study the interaction of various proteins involved in endocrine diseases and disorders. And our approaches are using Monte Carlo simulations as well as molecular dynamics simulations. All right. Then we also do a lot of innovative things in the lab, and one of them is developing low-cost microscopes for education, which can be assembled in 500 rupees. And we also develop robotic as well as non-robotic units for sterilization, and we have patented some of them. Now this is the promotion slide for the institute. Very recently, we have also set up a dual beam optical tweezer and magnetic tweezer, which is integrated with a confocal microscope. This is the first facility of this kind in India, and those who are willing to collaborate are free to approach us. So as said that I work on motor proteins and microtubules. The reason I like them because they are very important for cell division, where multiple microtubules and multiple motor proteins are involved in separating the sister chromatids into two daughter cells, and one of the very important thing that they carry out is the intracellular transport, which refers to the transport of different types of vesicles and cargoes inside the cell. So if you look at the cell, you'll find that vesicles are moving all the time in all possible directions. So you can think of that the cell is like a city, and these microtubules are like long range highways, and these actin filaments are like local roads. So anything which has to go from cell center to cell periphery has to take these highways, then local roads, and anything which has to come from cell periphery to cell center has to go in the opposite way. And typically there are two different types of motor proteins, kinesin and adenine, that move on the microtubules. And we are interested in this because the breakdown of this transportation system is responsible for various neurological diseases. So if you talk about the molecular motors, they are mechanical enzymes. That means they generate the mechanical force by hydrolyzing the ATP. And if you look at the real in vivo transport, then any cargo is not just transported by usually one single motor. There are a group of motor proteins which are involved in transporting a given cargo. Now, if you talk about the real in vivo transport is generally bidirectional. For example, many organelles like mitochondria, endosomes, lipid droplets move in both the directions. So if you tag any one of these moving, let's say, lipid droplets or any other vesicle, you'll find that it will run for some time, it can reverse its direction, it can pause for some time, and again can reverse its direction. So for a very long time, people were interested to understand what determines these different runs, reversal, and pauses. Now, very recently, people have also got interested in understanding how the motor proteins are arranged on these individual cargo surfaces, whether they are arranged in a dispersed arrangement or a cluster arrangement, and if so, what are the advantages of these different types of motor arrangements, and if any transition is possible between these two types of motor arrangements, and what factors might drive this transition from, let's say, dispersed to clustered or clustered to dispersed arrangement. So this is what we try to address. And this was something which was motivated by work of our own colleague, who is now our own colleague, Professor Roop Malik. And what they have found that, if you talk about the dynein motor proteins, they cluster into ensemble into lipid microtubules with phagosome maturation. So if you look at the phagosomes, when they are in the early phagosome state, or when they are in the early stage, you will find that dynein motors are dispersed, but as they mature, they cluster together, and then this phagosome generates a large collective force which drives this cargo towards the lysosome, where the pathogen can be degraded. And many pathogens like Leishmania disturb this microdomain, and they hijack this entire transport mechanism to avoid the to avoid and to infect more, basically. So the clustering of motor proteins on cargo surfaces have been studied by various groups using both in vitro as well as in vivo studies. For example, One group by Chaudhary et al. Studied the spatial distribution of dynamic clusters on small cargos and their cooperativity on smaller endosomes. However, the problem is that they did not quantify the relevant parameters for the collective transport, for example, run length and trajectories and velocity. Then there was another study by a group of Professor Jingju at UC Merced. They again looked at how the clustering happens, and what they found that the effect of motor clustering on the run length is limited by the stochastic tumbling of the cargo during the transport, and they also found that the sliding of the motors within lipid membrane delays the force-based interaction between motors and enables the leading kinesin to move forward further before the lagging motor dissociates. So this is what they proposed. Then there was another work by Nelson et al., and in this case they used the cargos with both fluid as well as gel type membrane, and they studied how the transport properties will be changed. So they found that the velocities are changed, but there is no effect on the run length. So there are few experiments, and there was no comprehensive study of what happens to the average transport properties of the cargos due to this sliding of motor proteins on the cargo surface. And then there were also some theoretical models. The problem with the theoretical models is that in all of these theoretical models, they consider the motors to be rigid one, and in the first model, they even work with only fixed arrangement of the motor proteins on the cargo surface. So what we wanted to do, we wanted to understand the effect of different motor arrangements and sliding of tails on the cargo surface, and to understand the effect of varying tail diffusivity under different forces and torques on drift and diffusion of these motor tails and their role in cargo transport by motor proteins in disperse as well as cluster arrangement. So what we did, we came up with 4 different types of arrangement. One of them we call is anchored disperse, where motors are anchored on the cargo surface in a dispersed manner. The second one is diffusive disperse, in which motors can diffuse on this cargo surface and they are randomly distributed. The third one with which we came up with was anchored cluster. So motors are anchored but within a given patch on a cargo surface, and in the third model, which we say diffusive cluster, motors can diffuse only in a given patch. So we had these 4 different models, and then we let also the motors diffuse in the models where we model the diffusion of the motor tails, and we did the simulations with total 10 number of maximum 10 number of motors. This is already published work, so I will not go into the details. So what I would like to add, that when you have a cargo which is being driven by the forces, then these forces can cause the cargo translation, they can also cause the cargo rotation. And what you have to do in the simulations, you have to basically update the cargo position, but before that you have to also update the motor head position, you have to update the cargo position, and then since motor tails are residing on the cargo surface, you have to also update the motor tail positions. So we did all of that. I will not go into the details of this. We solved the Langevin equation for that, both translation and rotational. And the quantities of interest for us are intermotor distance and cluster area, which we calculate, and we also calculate the cargo transport properties, which are run length, velocity, and cooperative index. And what we find, let me directly go to the results. So what we find, that the trends for run length, velocity, and cooperative is different for different motor arrangements. What we find, that anchor cluster performs much better than the diffuse cluster, and diffuse cluster performs much better than the diffuse dispersive, and the worst performance is shown by the anchor dispersed arrangement. And what we learn from these simulations, that diffusion dispersed arrangement does not change the number of motors attached to the microtubule, but slightly decreases the force acting on each motor due to flexible motor attachment points. Then diffusion in the cluster arrangement decreases the number of motors attached to the microtubule, which increases the force acting on each motor. And then we also included the effect of 3 different types of diffusion. We call 3 different types of torques acting on the motors in these diffusive arrangements. So we included the random torque as well as motor torque, and we also included the combination of random and motor torques in the arrangement where the motors were clustered in a patch, as well as in the arrangement where motors were free to diffuse on a cargo surface. I don't have time for discussing all the results, so what I will do, I will just tell you the summary, that what we find, that if you have to compare the diffusive and dispersed tails, then we see the maximum run length in the diffusive dispersed motor torque model. If we have to compare the diffusive and cluster tail model, then we see the run length is maximum for diffusive cluster model with the motor torque. And if we have to compare the performance of diffusive dispersed and diffusive cluster models with the combination of both random torque and motor torque, we see that the motor tails do not diffuse back to dispersed arrangement after forming the cluster, and at any instant tail keeps diffusing away due to thermal fluctuation, but restoring motor torques pulls them back together with a directional drift. So these are the summary of our work from all of these simulations. The run length, velocity, and cooperativity index improve with the diffusion for tails in the dispersed arrangement, but deteriorate with the diffusion for the tails in cluster arrangement. Random torque does not cause the clustering of the tails in dispersed arrangement and leads to sliding away of tails in the cluster arrangement. Then motor torque leads to motor tail clustering in dispersed arrangement and holds cluster tails together in the cluster arrangement. Run length is maximum for the motor torque model in dispersed and cluster tails. Velocity and cooperative index is maximum for the combination of motor torque and random torque model in dispersed and cluster tails. So thank you very much.